Perfect. See, this step was DNA separation from the liquid. Top layer liquid, supernatant you transfer. So we got clear liquid into the next tube. Clear? This is the good way to transfer, and you did wonderfully. Wonderfully. Even PhD student not able to do like that way. Because you listen to me and keep the silence, right? Right? So that is the good way to do this. Clear? You did wonderfully. I am very pleased to see your performance. Everybody show me the sample like this. Your sample is separated very well. Okay? So this is the fifth step. DNA after cell lysis, protein precipitation, and top layer liquid that containing DNA you transfer into the new tubes. And very well you did. Okay? After lunch we will see the DNA. Thank you very much, everyone.